We are the Roy sisters. Noemi is an MD and lives in Oxford in the UK, and Anna is an RN and lives in Brooklyn, New York. This is a very simplified video to help you process the information and guide you in making the right decision for you.
COVID-19 has been roaming the earth for a year now, taking our loved ones, greatly impacting our way of life. This past year has caused so much stress on us all. This month, the scientific teams of the world have finalized the production of some vaccines, with dozens more still in development. We have heard so much about the fight for finding the cure, but today, many of us are fearful as there has been lack of information. Let us take you into how our bodies amazingly protect us from attacks from viruses, bacteria, and fungi.
There are different types of defense mechanisms. The non-specific defense is composed of the first and second line defense. The first line is the skin and mucous membrane, which prevent anything from entering into the body.
If a virus, bacteria, or fungi does penetrate the body, this triggers the second line of defense, which is an inflammatory response. You've seen it when you cut your finger. On the left of the diagram is your bloodstream, where the white blood cells called leukocytes are floating around. The inflammation reaction causes the white blood cells to go to the site and swallow and dispose of the attacker.
If the attack is more complex, then our bodies have developed a more specific defense system called adaptive immunity. Let's focus only on the viruses. They have a small protein on the outer part of their membrane called antigens. These antigens trigger our immune system to find a specific, tailor-made solution called antibodies.
Once again, our white blood cells are called to defend us, but this time it's a different kind, the lymphocytes. We have two types of lymphocytes, the B cells and the T cells. B cells are floating around in the bloodstream until they find a trigger. They contain a Y-shaped protein found on the outer membrane of the cell called an antibody. It allows them to fix themselves on the antigen of the virus and mark them as being ready to be removed from the body. Our bodies spontaneously produce different mutations of B cells in order to potentially match any type of attack. The T cells are divided into two subgroups, the helper T cells and the killer T cells. Helper T cells stimulate B cells to make antibodies. Killer T cells kill cells that have been infected by the invader.
The advantage of adaptive immunity is that the immune system can learn and remember specific pathogens and can have long-lasting defense through antibodies. This is a memory cell that has a copy of the antibody. If one day our body comes into contact with the same disease, we can produce many antibodies to defend ourselves and not even get sick.
So today we will be focusing on how our bodies defend us from the COVID-19 virus.
Viruses are tiny particles that hijack our cell's genetic material, the DNA, to produce replicas of itself. This is an actual representation of the COVID-19 virus. Can you see all the proteins found on the outer membrane of this virus?
Now, these past few months have slightly been chaotic on understanding how COVID-19 can attack our bodies. We have seen how it can impact multiple organs differently depending on the person it uses as a host. You can pause the video here if you would like to read all the different ways COVID-19 can impact your body. It can range from basic symptoms similar to the flu, just a loss of taste and smell, to clotting issues, respiratory issues, or you may sadly lose the battle. If you're infected with COVID-19, you will not know how it'll impact you until you get it.
Today, we have reached a point when several COVID-19 vaccines are available. It's been interesting to watch how the scientific community throughout the world has decided to face finding a solution to COVID-19. There are many different types of vaccines.
Vaccines are introduced in the body to mimic an infection and cause the body to produce an immune reaction called antibodies to fight the attack. Up to now, most vaccines were either weakened pathogens, this means a damaged version of the real bacteria or virus, like the vaccine of the measles, mumps, rubella, and chickenpox. The pathogen is weakened as to not cause an infection, but it is also so similar to the natural infection that the immune reaction is very strong and lasts a long time. The second type of vaccine is called an inactivated pathogen. In this version, the ineffective agent has been rendered completely unable to multiply and attack. This is the case for the flu shot and polio shot. The immune reaction is not as strong and may need booster shots. The downside is that although these viruses and bacteria have been weakened, they can still multiply and can cause severe reactions in people with a defective immune system. The third type is a recombinant vaccine, where a small piece of the virus is created in the lab and then injected in the body to create an immune reaction. In this case, the virus that is used for the vaccine is incomplete, so you can never cause an infection. This is the case for hepatitis B, HPV, and shingles. The immune reaction is strong and can be given to anyone, even those with a weakened immune system. The downside is that you may need booster shots to maintain immunity.
There are two newer types of vaccines that don't fall into any of the categories explained on the previous slide: the messenger RNA and the viral vector vaccines. The first type is messenger RNA, or mRNA. Messenger RNA is used to copy protein in our bodies. In this scenario, a strand of mRNA is introduced into the body to encode for the creation of the protein antigen to create the immune reaction. In other words, your body will build the antigen from the mRNA that you will receive. You are not receiving any part of the virus. It is all synthetic to encourage the body to create antibodies. The RNA is placed in a small capsid to be able to be injected in the body. Although this is a new form of vaccines, mRNA vaccines have been injected in humans in the past to treat other conditions and found to be safe. Many people worry that this may mix with their own genetic material and could be passed down to future generations. This cannot happen because mRNA is different from the DNA that is in your cells and passed to your children. mRNA is just the messenger that takes information from DNA to proteins. It is not long-lasting and it gets destroyed naturally.
The viral vector-based vaccines inject a genetically purified spike protein from the coronavirus gene to the body using a genetically engineered common cold virus as a middleman to carry the gene into the body. The body then produces the spike protein, which stimulates the production of antibodies. This is actually an old technique that dates from the 1980s with the dengue virus.
There are currently 4 vaccines about to reach their final stages of test and to be given in the population: AstraZeneca, Johnson & Johnson, Moderna, and BioNTech Pfizer. AstraZeneca and Johnson & Johnson are both viral vector vaccines, and Moderna and BioNTech and Pfizer are the mRNA vaccines.
In order to produce a new drug, very rigorous steps need to be followed. Once the drug is ready, it can be tested on people in clinical trials. There are 4 phases to a clinical trial, but for COVID, only 3 phases were tested to this day. Phase 1 are small, less than 30 patients. The goal is to find a safe dose, determine how the treatment should be given. Then in Phase 2, there are more patients tested, about 100, for a longer period of time. It explores how it affects the body. New combinations of the drugs are also tested at this stage. Phase 3 requires more than 100 and at times thousands. It explores the side effects and standard of care.
These are the trial sizes of the 4 vaccines that we're exploring and are following. AstraZeneca is 30,000, Johnson & Johnson initially aimed for 60,000 but carried out 40,000, Moderna was 30,000, and BioNTech Pfizer initially aimed for 30,000 but retrieved 44,000 candidates.
Okay, that's enough information in one gulp. Let's just explore a few questions that you have been pondering.
Why did this vaccine come out so fast?
Some of you may wonder why the vaccine came out in record time when the fastest vaccine ever produced before this one was the mumps vaccine in 4 years. Researchers led by Dr. Wang Jianwei at the Chinese Academy of Medical Science Institute of Pathogen Biology in China were amazing to publish the genome of COVID-19 on January 12, 2020. This enabled scientists from all over the world to begin working on the vaccine before the first wave ever hit Europe or the US back in the spring. Importantly, many resources all over the world were poured into making this vaccine. So when there is not so much urgency, a lot of delay in getting a new vaccine will be the waiting time. Time for scientific teams to apply for grants, time for the grants to be reviewed, time for the funding to be awarded, for the staff to be employed, etc. In this case, all of these steps were cut drastically short so that people could focus on just the vaccine. Other work in labs shut down, and many more scientists focused on developing the vaccine than would usually be the case.
In addition, many teams were already working on coronavirus vaccines, so had everything set up and could start as soon as the sequence was published. To save more time, some of the clinical trial phases were carried out simultaneously, shortening the time needed for each phase. BioNTech was already a leader in flu shot vaccines and already had the techniques ready to make mRNA vaccines. We also saw that the viral vector vaccine was a technique already used back in the 80s. An mRNA vaccine does not require much time to produce. A simple algorithm is produced and the genetic sequence is simply encoded in the software, making it an easy production. There were very large investments in the business to find a rapid solution, and a major incentive as the COVID-19 virus was very virulent and killing many. Lastly, the FDA accelerated the approval of the vaccines early, simply because it was a public health emergency. It doesn't mean that they spent less time analysing the data. It means that they stopped looking at any other drugs and put the vaccines at the top of the queue and reviewed them first. None of the steps on safety were actually shortened.
What is the efficacy of the vaccines?
The AstraZeneca vaccine has a reported efficacy of 70%, Moderna 94.1%, and Pfizer-BioNTech 95%. We're still waiting on the results from the Johnson & Johnson vaccine.
This diagram highlights that we already accept other vaccines with less efficacy. Also, 3 vaccines, the AstraZeneca, Moderna, and Pfizer, will all need 2 doses 3 weeks apart, whereas the Johnson & Johnson will only require 1 dose. It is difficult to administer 2 doses and to have patient compliance. We do not know the long-term impact, nor how long the immune protection will last.
How long does it take for your immune system to reach immunity? After the first dose, your body has reached a 50% immunity. 3 weeks later, when you receive the second dose, it takes your body another 2 weeks to reach maximum immunity.
What are the advantages and disadvantages of the vaccines? Viral vector-based vaccines are well-established technology, have a strong immune response by stimulating both the B and the T cells, and create a robust immune reaction without the use of an adjuvant. The messenger RNA vaccines are safer because they are produced in a cell-free environment, do not enter the nucleus or alter the DNA, and also stimulate T killer cells. They are easy to produce, and any protein can be encoded and expressed, even when the virus mutates.
Viral vector-based vaccines may have a reduced effectiveness if there has been previous exposure to the virus. They are also more complex to manufacture. mRNA vaccines are more fragile and more difficult to deliver to the cell. They require a booster shot and to be maintained at very low temperatures of minus 94 degrees Fahrenheit for Pfizer and minus 4 degrees Fahrenheit for Moderna.
What are the side effects of the vaccines?
To this date, there have been 8 cases of people receiving the Pfizer-BioNTech vaccine and having an anaphylactic shock reaction. 2 in the UK and 6 in the US. Anaphylactic shock can manifest itself as rash, a plummeting blood pressure, shortness of breath, and a fast heartbeat, and usually occurs extremely quickly after the vaccine is given. The cases are being investigated but were all resolved after appropriate medication was administered by the staff giving the vaccine. It is believed that the reaction could be due to a nanoparticle in the packaging of the mRNA strand, and this same nanopackaging is used in the Moderna vaccine. It's also important to remember that anaphylactic reactions can occur with any vaccine, indeed any medication that is taken, but they are usually extremely rare. Unfortunately, we do not know of long-term impacts of these vaccines. They are considered very safe, and we know that no genetic material is being altered.
What are the counterindications of the vaccines? Speak to your healthcare provider about your health conditions to make sure that you can and should take the vaccine. Clinical trials were tested on adults from 18 to 65 and not on pregnant or breastfeeding individuals. Also, people with previous reactions to vaccines should take care. We hope that this video has helped you gain a clearer understanding of the different types of vaccines being offered at the moment. With the help of your healthcare provider, you can weigh the advantages and disadvantages of both the potential reactions you could have if you were infected with COVID-19 versus the potential reactions of receiving the vaccine. We should be thankful and feel privileged to have devoted and competent scientific teams able to collaborate and bring forward vaccines in such a short time frame to help us bring back a sense of normality in the year to come. We believe that the information is accurate as of end of December 2020. Following are our references that you can also find cited below in the comments section of YouTube. We welcome comments to improve this video. Thank you for having listened.